Hello everyone, this video has discussed about red rot of sugarcane, the main pathogen that attacks it, the occurrence of the pathogen, the mode of spread, the symptoms, the disease cycle, and lastly the control measures. Red rot of sugarcane. Red rot of sugarcane is a type of disease that is seen in the sugarcane plants when a specific type of fungi attacks to it, and that specific fungi is, we will do about the fungi in a while, but what actually the fungi does is that it attacks the sugarcane plant, and because of the attack by the fungi, the plant starts to undergo certain changes, and one of the major changes that occurs inside the plant is development of reddish areas around the vascular bundles, and these areas are produced in huge amounts. That is, the reddish areas are produced in huge amounts. Also, reddish area starts to appear on the upper surface of the leaves along the midrib, and these reddish areas are bad. These are literally very bad because they tend to cause many economic losses. How? The question is how? Well, actually it has been seen that these reddish areas, when these reddish areas are present, it tends to change the natural taste of the sugar. That is, the taste gets reduced, as well as the quantity of sugar that is produced from such diseased plants are also less, so the yield is also less here. And again, it has been found that the seeds that are produced from such diseased plants are of very poor quality. So now, what is that fungi that we are talking about since the first slide? The name of the fungi is Glomerella tucumenensis. Earlier, this fungi was used to be called as Coleotrichum falcatum. So Coleotrichum falcatum is the older name, and Glomerella tucumenensis is the new name, and this name is in use at present. And this is the fungi, Glomerella tucumenensis, the pathogen basically, that causes the red rot disease of sugarcane. Now, when scientists isolated Glomerella tucumenensis from the diseased sugarcane plants having red rot disease and observed it in microscope in lab, they found out that Glomerella tucumenensis is a mycelial type of fungi that is made up of or is composed of many hyphal networks, and each hyphae was found to be multicellular and septate. That is, the cells were separated by common walls. Also, it was found that the fungi was very profusely branched. That is, very high level of branching was observed in this particular fungi, and had characteristic oil droplets. Suppose these are the oil droplets, so each cell had some specific oil droplets present in them. Now, as the fungi grew more, branched more, and developed more and more, it was observed that certain tiny blackish structures started to appear in the hyphae of the fungi. These tiny structures, blackish structures, were called as acervulus, and it was found that when these acervulus were zoomed in the microscope, they kind of appeared like this. Inside these acervulus were found to be present certain conidiophores. The conidiophores are closely packed, compactly arranged. Inside the acervulus are short, hyaline, single celled, and unbranched. That is, inside these acervulus, certain structures called as conidiophores were present. The conidiophores arise from the basal cells. The conidiophores were elongated, they were single celled and hyaline. That is, the appearance of the conidiophores was somewhat colorless or very lightly colored and had a very classy appearance. Also, it was found that the conidiophores were unbranched, and the conidiophores were found to be present very close to each other, and the conidiophores were present in large number. And usually a single conidium develops at the tips of each conidiophores, but occasionally chains of conidia gets formed. That is, at the tips of these conidiophores, a single conidia was found to be formed. You can see here, these are the conidia, and the shape of the conidia was kind of sickle shaped like this, and the conidia were uninucleate. It was found that during certain conditions, chains of conidia also got formed, but this was rare. And again, it was found that because of certain abiotic conditions like rain or wind, these conidiophores got detached from the conidiophores and got spreaded. For example, here you can see maybe these conidia which are present here in the above portions, they have got detached from the conidiophores. This is a conidiophore, this is a conidiophore, all these structures are conidiophores. Now, it was observed that not only structures like acervulus were found to be present in the mycelium, but certain additional structures like perithecia were also found to be present within the mycelium. The perithecium are again Perithecia is a plural word and perithecium is a singular word. So these peritheciums are globose shaped, and again inside these peritheciums spores are formed, and the spores are called as ascospores. These ascospores are present within asci. So this is a picture of perithecium. Inside the perithecium are present asci or ascus, singularly ascus, and when talking about plural, that is many ascus are considered, it is called as asci. So inside these asci or ascus, 8 ascospores were found. So these are the two types of spores. Also, another type of spore called as chlamydospore was also found in the mycelium. The chlamydospores were roundish in structure. Now these two diagrams, acervulus and perithecium, is not the very complete diagram of what acervulus or perithecium basically looks like, because the complete diagram is very complicated. For example, in the acervulus, apart from the conidiophores, the conidia, certain structures like seta also appears, which are needle-like and very much elongated. So making the whole picture would have made it the acervulus very complicated. So I have just mentioned those parts which are actually necessary. That is, just I have shown the spore portions. Again, in the perithecium, apart from the ascus, there are also present other certain structures that can be seen if you just type in the Google and see. Many acervuli, many perithecium, many chlamydospore develops in a mycelium. Now let us study about the occurrence of the pathogen as to what are the conditions that favors the growth of the pathogen. First is warm and humid weather conditions with intermittent rains, that is warm climate, warm soil, warm atmospheric conditions, as well as humid weather, humid conditions favors the growth, and intermittent rains, that is small or very little rains. Optimum temperature is 28 to 30 degrees Celsius. The pH range at which the fungi works very efficiently is 5 to 6. The fungi prefers high atmospheric humidity, about 90%, and waterlogged condition is acts as a boost for the fungi. It has also been found that drought conditions during the initial stages of infection by the fungi causes a very rapid increase in the growth of the fungi, as well as monoculturing of sugarcane has also been found to increase the growth of the fungi. Monoculturing of sugarcane means when a sugarcane plant is cultivated season after season after season. Mode of spread, that is how the pathogen gets spread. One very common method by which the pathogen gets spread is by farm equipment and by abiotic factors and biotic factors. Abiotic factors like rain, splash, wind, etc., and biotic factors like insects, animals, humans. There are certain specific symptoms that occur because of this fungi. So what are those symptoms? So basically it has been found that initially the visual diagnosis at the early stages of infection is difficult to recognize. The reason here is because the fungi, after it reaches a plant, enters a plant, it first attacks the stem, and that too the internal portions of the stem, and mainly the vascular portion, the xylem, the phloem, and it grows inside the plant and spreads inside the plant, spreads inside the stem, and external symptoms do not appear. It is only when external symptoms starts appearing, like symptoms in the leaves or in the external portions of the stems, that at that time we can recognize it. But before that, we cannot just see a particular sugarcane plant and say whether it is a diseased plant or a normal plant, because externally everything looks very normal. The symptoms appear on the aerial parts of the plant. So first the symptoms appears on the aerial parts of the plant. Initially, as well as later on also, the symptoms mostly appears on the aerial parts only. Discoloration of the leaves is the first symptom that is seen in the field. So first symptom that by which one can detect as to the plant is infected by glomerular trichomonasis is by looking at the leaves. That is the first symptom, first external symptom. Now, what are the symptoms that are observed in leaves? And there are certain symptoms that are observed in the stalk, that is the stem portion. So the symptoms that appear on leaves are reddish lesions. Lesion means areas. Reddish areas starts to appear on the upper surface of the leaves along the midrib of the leaves, with dark spots in both the direction of the upper surface of the midrib. The lesions are initially blood red with dark margins and later on become straw colored. That is, suppose this is the leaf, and this here, suppose this was the midrib. So along the midrib, redness starts to appear, reddish area starts to appear, and the redness occurs in both the direction, that is in this direction also and in this direction also, that is towards the apex as well as towards the base, and the redness is along the midrib. After a while, when the infection becomes a little severe, from in the middle of the um, reddish area, the middle of the reddish area starts to become a little darker in portion and gives a appearance of a straw color. And this occurs because we learned that conidia starts to form, and the conidias are black in color. And because of this reason, a very because of the dark nature or blackish nature of the conidia, this portion starts to appear a little darker. So that is what has been said here. Also, yellowing and drooping of leaves starts to occur because now the plant is not able to synthesize the right amount of nutrition, as well as is not able to photosynthesize properly and have a very dull appearance. Drooping of the leaves occurs because the vascular bundles have got affected, because the fungi is growing in the vascular bundle. So water is not able to reach the leaves in a very high quantity as it was used to reach when the fungi was not present. The symptoms that occur on the stalk, that is the stem, are shrinkage of internodes. Shrinkage means shortening of the internodes. In a normal sugarcane plant, the internodes appear somewhat like this, they are elongated, but when a disease, that is when pathogen glomerulus tucumenensis attacks it, the internode starts to shorten, suppose this much. So you see, initially the length of normal internode is this, but the diseased internode is this much. Next is when the cane is split open lengthwise, red blotches appear on throughout the length of the cane around the vascular region. That is, suppose this is the stem of sugarcane plant, and now if the sugarcane is cut longitudinally, that is in this direction, suppose in this direction it has been sliced in this direction and then cut open, so it will look somewhat like this. A normal sugarcane would have looked something like this. The interior portion might have looked very whitish in nature, but now because of attack of the fungi glomerular tucumenensis, the internal portions around the vascular bundle appears reddish in the internodal portions, and the blotches or the areas run lengthwise. Also, it has been found that the infected cane emits a acidic sour smell while its juice emits a alcoholic smell. That is, these reddish areas, when the condition becomes severe, these reddish areas tend to emit an acidic smell as well as alcoholic taste and smell. And the reason as to why this happens is because the sucrose, initially the sugarcane plant stores sucrose inside it, but because of the attack of fungi, the sucrose gets converted into glucose and alcohol, and it is because of this reason that they appear so that the smell appears so acidic and the taste appears so alcoholic. Now next is the disease cycle. So the disease cycle basically starts with the attack of pathogen in the sugarcane plant. So initially the conidia or the chlamydospores, they attack on different parts of the plant. Basically what happens is that these conidia or chlamydospores via abiotic and biotic agents reaches the plant surfaces, that is the sugarcane plant surfaces, be it the bud or the leaf or the node or the stalk, that is the main stem. And after reaching there, they start entering inside the particular plant by releasing certain chemicals. So the spore germinates, suppose this was the spore, now the spore germinates, forms a little hyphae, the hyphae secretes certain chemicals and breaks the cell wall, dissolves the cell wall, the protective layer of the plant, and starts entering inside the plant. That is what has been written here, spore germination and entry of the hyphae into the internal tissues. Now once it enters, the mycelium spreads cell to cell in the intercellular spaces as well as intercellular spaces. Suppose these are the number of cells, so the mycelium spreads either by crossing the cells or it starts growing within the intercellular spaces, and it starts growing or spreading with the help of vascular bundles, that is the xylem and phloem. And after the mycelium starts spreading and developing and growing, then later on it starts forming acervulus, perithecia and chlamydospores. Now after these structures gets formed, the fungi starts attacking. The fungi also starts taking nutrition from the plant, the nutrient, the sugar molecules, everything from the plant, and because of that reason reddish area starts to appear in the vascular portions of the plant as well as in the leaves along the midrim that we saw in the slide of symptoms. Now, two things can happen after this. The first thing that can happen is the spores that are present on the leaves here, they via certain abiotic agents as well as biotic agents can get spreaded and then again attack different types of plants. I mean different types of sugarcane plants present in the field as the same plant or maybe the different plant. The second thing that can happen is these infected parts might fall in the ground and after that remain in the soil as plant debris, and or maybe the whole stem, the whole plant, if left in the field as it is, they may remain inside the plant as well and may remain dormant and overwinter. That means that undergo a period of dormancy until the next season, the new season comes, and when the new season arrives they break their dormancy and then these spores affect new plants which are now present in the field. So this is what the cycle basically is, and the cycle continues on and on and on and on. Now here there are two terms, secondary inoculum and primary inoculum. I'll be discussing these two terms separately in a separate video which I'll post very soon. So be tuned and please watch that video. Now here just two types of spores have been mentioned. One is conidia and the other one is the chlamydospore. But in the first slide we saw that there are three types of spores that are formed by Glomerella tucumenensis, conidia, chlamydospore and ascospore. But at present scientists have not been able to find as to what is the role of ascospore in the disease cycle. So this is still a question mark at present. Lastly, the last slide is about control as to how the disease can be controlled. So a very basic step is field sanitation and regular inspection by observing daily regularly as to if any symptom is observed, if any weird type of symptom is occurring in any type of plant, that plant first should be checked, and after that if it is suspected to have a disease, if it actually has a disease, it should be removed from the field carefully and then it should be burned far away from the field. So that is what has been written: removal and destruction of the infected plant debris, stubbles and trash. Avoid flood irrigation. Why flood irrigation should be avoided is because the leaves contain spores, and if these leaves falls on the ground and after that flood irrigation is present there, then the spores can be carried with the help of irrigation water to different places and can spread to a large number of area. The next thing is ratoons of sugarcane should not be kept in the field having red rot disease. These ratoons should be carefully taken out and after that removed from the fields. Seed treatment should be done like hot water treatment or by treatment with certain fungicides like arazan. Crop rotation with non-host crops like rice should be done for few seasons, maybe 2 or 3 seasons, and resistant varieties should be preferred. Many examples have been provided here. So this is it for this video. Have a good day, take care, keep smiling, and make sure to subscribe Core Botany, and if you have any doubt, do mention it in the comment section.